Welcome, everyone, to the Diatom Web Academy. I'd like to say that, well, before I announce today's speaker, in 2 weeks, Tuesday, February 28th, we will be talking about the Diatom Level 2 certification, which is a species level certification exam. And the Diatom Taxonomic Certification Committee, who also developed this Web Academy, will be presenting that session. So please join us again in 2 weeks. And for today, I'm delighted to have David Harwood, who is a professor at University of Nebraska. He's the Stout Chair in Stratigraphy. His area of expertise is primarily in Cenozoic and Cretaceous paleo environments. He works primarily in high latitudes, and I always knew David's work from the Antarctic until just a few months ago. I learned about his work in the Arctic as well, and looking at diatoms under the Greenland ice sheet, which was amazing. So David uses diatoms primarily to look at paleo environments, whether that's climatic change, changes in marine productivity over big time scales. And today he's going to be talking to us about some of those very old diatoms. So David, thanks so much for joining us today, and I'll turn it over to you.
Yeah, sorry. Thanks everybody, and thanks for that nice introduction, Sarah. It's nice to be here to be able to share part of my world with you and take you to meet some old friends of mine. Not just that they've been around for a while, but they do go way back in time. And I'm just thinking about this name of Great-Grandmother Diatoms for this talk. I just want to think about our ancestry, right? And the diatom's ancestry as well. So we're going to be taking a bit of a trip through time, maybe 100 million years or so back in time. And I know that many of you don't think in this long geological time scale. So I think I just want to put a face on some of these diatoms that gave rise to most of the diatoms we have today, working primarily here within the marine record, but a few comments about some of the non-marine things coming in there as well. And I also want to review some recent research that's come out and some that's also kind of pending or planned, so you can get a sense about where some of the questions are taking us in terms of how we're going to address some of these things. And my slides are not advancing. That's odd. Hold on.
Sometimes it's the down button and sometimes it's the side button. I always have sort of a weird experience with advancing too. I see your mouse.
Yeah. Well, I'm going to stop sharing and start sharing again. Okay.
Okay. We've got a little technical going on, everyone, right now, but hopefully we'll get that worked out.
Sorry.
No.
Can you try clicking on the PowerPoint presentation? It might cause it to advance.
There we go. How's that?
Got it. Yeah.
Okay, good. Thank you. Whoever your voice from up there was. Thank you.
You should be able to use your arrows now.
All right. Thanks for that. Yeah. So here's just a look at some of these deeper time diatoms, and you might say those don't look anything like what I'm used to looking at, and probably not. But some common characteristics of these guys spanning the time interval from about 110 million years ago to 65 million years ago. I'm going to stay within the Cretaceous here for this discussion, but kind of focus on some of the old things. They have some kind of unique characters in that some of them don't really have what we would refer to today as a valve face, like many of these others. It's just the diatoms are just long cylinders, where most of the interaction with the environment, is through the areoles on the valve mantle. But I also want to acknowledge a number of people here who have helped within this type of project over many years, and some folks that are still working today. But in looking at diatoms from deep time, there's some special approaches we've been able to use and need to use to enhance their recovery. Some of that is with stratigraphic drilling, like with the ocean drilling program or the International Ocean Discovery Program, targeting some sites that are buried very shallow burial, so that they're not undergoing too much heat and pressure and changes, because the opaline silica that makes up our diatom valves and girdles and frustules is not stable through time. The opale will dissolve generally. And sometimes when there's replacement of the diatoms with pyrite, it can be just really ugly, but sometimes there's a very fine scale pyrite replacement as well, and that's just an iron sulfide that replaces the silica for that. But a real big step has come within looking at time capsules, in part where the diatoms within the sediments have been preserved or encased somehow within coprolites, chiefly of vertebrates, marine vertebrates, fossil fecal material, if you will, but they cement up pretty quickly. Or looking at diatoms within the sediments that are inside macrofossils, or sometimes when the macrofossil bodies will decay, they will lead to the precipitation, the pretty early precipitation of calcium carbonate in and around the sediments. And that will also help kind of ice the diatoms up, if you will, put them in a carbonate cement. So you just take those samples, put them in hydrochloric acid, they'll break down, and they will have been protected from the diagenesis of time. A new occurrence that some Japanese colleagues have identified was by looking at methane seeps, marine seeps, where methane's coming out, carbonate will be precipitated there as well, and is an enhanced way of recovering some diatoms. And then French colleagues here by looking at diatoms in ancient tree sap from material that's blown onto the trees on the coastal environment and capturing whatever happens to be in the sea at that way. But one thing is certainly clear, that serendipity has played a role in providing us with new occurrences. Here's an example of some of the material coming from amber. And the beauty of this as well is it's almost, right, like getting the living beasts, right, that are blowing just from the water onto the tree sap and then being preserved, so that you're still seeing them in colony formation and chain formation like this. So a fruitful area for seeing diatoms that never made it actually into the direct sedimentary record, right, if you will. Some other diatoms, these are some non-marine ones coming from Mexico, that they're being preserved in chert. So chert's like a microcrystalline kind of quartz. And you can see in here also preserving colonies, non-marine things in here, and also another way to look at these in a I don't know if I'd say it's definitely like a near life position or a Lagerstätten type of thing with excellent preservation. But I also want to give you a sense of what is the nature of our record, and it is getting better and better and extending farther and farther back in time. So this is just a plot of most of the deposits that are out there and their distribution through time and their distribution across latitude through here, from equatorial out to the different polar regions. And you can see that for the Lower Cretaceous time period, 120 up to maybe 100, we don't really have an awful lot of sites. Many of them are in the southern hemisphere, and they're chiefly from hemipelagic or a clastic sediment component there, where you're on a continental shelf. Probably you're close to sources of sediment coming in. And then when we go up into the Upper Cretaceous through here, There still are some of those, coastal or continental shelf settings, but we start to see for the first time that the type of sediments are going more towards an oceanic realm. We actually see things that look like that are diatomaceous rich. So whether that represents the diatoms were, closer to the shelf, closer to the coastlines, and then they've made their way out into the open ocean, may have been something that's a progression for exploiting that planktonic habitat directly. And this is a figure that just shows where many of these sites are located. I would have to add a few onto here to update this a little bit, but you can see spanning from both polar regions and some in the equator as well. Although in some cases, preservation can be a bit of a problem, as you will see. Here's a kind of a summary figure that is pretty old by this time, but something that Vladimir Nikolaev and I put together to explain a contrast between these early or lower Cretaceous diatoms, which are some work that Vladimir and Rainer Gersonda and I described, kind of very fortuitous occurrence from a drill core, and then what we understand a bit better, longer record of study of these things from the late or upper Cretaceous. You can see a lot more genera, right, are coming in compared to these. All of these genera, most of these genera were new from the work that that Rainer Gersonda and I and Vladimir Nikolaev did. But what I want to point out in this is you can divide these up. This appears to be a turnover. My PhD student, Megan Hines, refers to this as kind of the diatom dark ages, because we previously did not have any good information coming from this time interval. We had a fair bit from the Maastrichtian and Campanian, like the last 85 or so million years, some spotty occurrences through here, and then some good occurrences down in this time interval. But that transition between this early phase and then like a middle phase of diatom state of presence there, we're lacking some information, and there appears to be a turnover or a break in these, where these guys go away, disappear. Doesn't appear to be a very abrupt extinction, and then some of these appear to be giving rise to all of these other genera up here, and particularly two in here seems to be a bit progressive. This one called Trochus, which I'll show you a little bit later. It appears to be one of the few in this time interval that has primitive type of labiate processes here. Or Bilingua, the only genus in this older occurrence that is like has two poles on it, and it has a ring of labiate processes. So it's likely that these two gave rise to many of those groups that you see up here. So there's kind of some connection between the lower part of this figure and the upper part, a little bit loosely. And then over here, we have some resting spores and some other groups that are not treated so formally yet. But I also want to keep in mind of other potentials for looking back into the past and seeing how diatoms are organized. I didn't put a reference on here, but this is from Kenta Abe and Rick Jordan and my work on a group of Corythron, early Corythron things down from here. And this is a tree that Matt Ashworth pulled together. But it was really useful to identify or put some of the older genera, even some modern genera here, like Leptocylindrus or Stephanopixis and Archipyrgus close to Aulicosyra through here, and just see where these genera will fit within these trees. But then also trying to balance this in some way by thinking about a loose assemblage of maybe time, going from older up to younger with these different branches here, and also to relate within some of those important diatom records that we have of where many of these splits, right, or these branches may have occurred. So in the Devon Island material from the Arctic, we see Leptocylindrus right through here in the what LC 113 stuff, and on Devon Island in the Canadian Arctic, we start to see ancestors of some of these here, Melosyra or Paralia. But you can see that if we were to be targeting taxa that are still alive today, for some of this, we can see many of these that are dominant within these really, really old records. Just kind of a cross between molecules and stratigraphy here, and then to just see how things play out So this approach to looking at great grandmothers of diatoms really took a big step forward in the late 1980s when ocean drilling program drilled off the Weddell Sea margin of Antarctica and recovered these really pristine diatom materials, as you can see, really well preserved. They were in black shales, and they gave us a pretty good look at what was going on with diatoms back in this time period. Many of them have these long paravalvular axes, right? Many of them have strong linking spines, linking connections in this with direct physical connections. They form chains. They had resting spores associated with many of these, and they were all relatively heavily silicified. One other thing that they all seem to represent at this time is that they had kind of one basic plan for adding silica onto their basal siliceous layer, if you will, because the nature of the pores, their spacing, their size on the valve face, and which continues underneath these secondary strengthening things, and even on those that are just single skin without these secondary pseudoloculae, or even the nature of the girdle bands on some of these. Like they had this a single way of adding that silica, and then they are modifying that in some ways. But they all had that common radial perforate silica those early times. You can see that in some of these here that also did not develop that loculate structure. It was all simple radial perforations. You can see they're all pretty much the same size. This is trochus. These are probably early forms of the labiate process. Here's bilingua, the only, again, bipolar type of diatom at this time, and there are these radial slits represented through here. There are only 4 specimens of this genus known, so we either haven't hit his environment or it was still, pretty isolated somewhere. Many of these forms do show a marginal ridge of some sort, and they also show this nature of a radial costate coming off the valve face, bifurcating into these marginal costae, and a spine being present then at the margin of that contact. So many of those types of diatoms I just showed are present down in this time. Another interesting group that seems to be a pretty dominant group within many of these old assemblages from Antarctica are in these resting spore groups, and I want to focus on Calyptosporium here. Kind of an interesting, abundant group, pretty diverse as well. And there's likely a connection between this as a resting stage or resting spore stage with this other diatom that is kind of like a rhizosolinia, right? Which probably had a very long girdle bands, representing much of the valve or the girdle through here. But these seem to be perhaps connected, and I'll show you how that works in a little bit. But I want to focus on Calyptosporium because it also gives us a link to an even older group of diatoms, or things we think are diatoms. So here's a sketch of Calyptosporium. It has this outer, you could call it an epivalve, if that term still works when you add multiple other parts to the valve. A hypovalve in here, perhaps, which is fully enclosed within this epivalve. And then another structure which most diatoms don't have today, or centric ones don't have, which we named an infundibulum, which means like a funnel, right? Which is on the end of this. These spines that are coming around here, they seem to wrap up on the inside of this infundibulum. The function of this is still not so very well known. You can see many of those spines coming in, and here they are lapping up over top of this funnel. Some other examples of that. And here you see the different nature of this epivalve through here, whether it has a ring or a crest or marginal ring, whether there are radial costae across the top, or whether it's covered with just some spines across that. But here's the nature of this internal infundibulum here, and get a sense that there might have been some kind of a plug across through here, or maybe this is just broken. Or you could even think that these spines are fine enough that they're actually flexible, right? That might be holding some things in place, or could even act in some ways like a spring. But it's these guys, Calyptosporium, that gave us some insight into a really, really old diatom from the Lower Cretaceous, maybe from 130 to 140 million years ago. And it's represented by these specimens you see here, which is a solid silica, kind of a helmet, if you will. And then in the bottom side of this helmet, you've got these discs, which are also bifurcating at the margin with a radial costate in here. And if it weren't for knowing what Calyptosporium looks like, we would not have been thinking that these, were related. It's amazing that this material from Korea even exists. It is actually coming from a metamorphic rock, right? But it's a calcareous metamorphic rock. It's cemented with some calcareous material. It used to be a sediment, but it's partly metamorphosed, which is why these things look so ugly. But thinking this might be, one of the earliest type of diatoms, but it is a non-marine diatom based on the paleogeography, right? And if you look at Japan, and then you look at Korea, and you look at China in this reconstruction geographically, you have marine material in Japan, you have terrestrial material in China, right? And it appears that this would fit within for these things to be non-marine. But still some things to tease out of that. And many of those valves, do seem to match some of those features from these earliest 100 million year, 120 million year old stuff from Antarctica. We kind of divide some of this for discussion purpose only into those diatoms that may have given rise to diatoms, or those groups that may have given rise to diatoms, the very early ones within this ancestral type of terminology, or within this time interval from Lower Cretaceous through here, maybe calling that more archaic, because there appears to be a group that was living at this time. And then after this transition, maybe associated with a major marine extinction here of the Cenomanian-Turonian boundary, where many marine organisms show extinctions across there. This is also within the time realm of ocean anoxic events, whether that would play a role in diatoms as well. But then stepping into this much better known, more diverse group, which we call medial, and then stepping into the Cenozoic, maybe from an Eocene time on, being in more of a modern type of diatom world. There are some araphid pennates, coming in around this time interval, which then may continue up into younger times. Also looking at some material from calcareous concretions from Queensland, Australia, put a different look on what diatoms were doing back in the day, in that some of these diatom records are in the form of colonial filaments. You can assume these might be benthic. The strata that are preserved in Queensland are reflecting times when the sea levels were rising and falling. Repeating phases of marine transgressions across Australia give us part of this record. And this material is pretty poorly preserved, but amazing to be able to find these long filaments, these long tapering filaments. If any of you in the more modern world know of diatom filaments like this and colonies that taper towards an end, I'd be interested to hear about that. But you can clearly see in the density changes from some of these filaments that there are cells, right, beneath what would appear to be an outer layer, whether that's a series of just poorly preserved girdle bands. You can see, some of that covering going across through here, but you can clearly see something that looks like it could be a valve, and a girdle, a valve, and a girdle on some of these things, and direct connection between the sister valves here. Right, so you get a sense from some of that. Again, the Australian material is pretty poorly preserved, but if we compare that material with the better preserved material from Antarctica, you can see how some of these associations might exist. Some of these diatoms get really large, right? So these are probably the same taxon. These are probably all the same taxon. It's just a change in size, right? That is giving us these different views. But in these colonial filaments, there are also these oddities over here, which, these look like two valves, like some, girdle bands or something covering the outside of those. But then there are also these cones or these tapering bits, which kind of look like that. So why on a filament like this would you have two different morphological expressions? Why would you have those things that look like, like hemispherical domes, which would be valves, and then they go into this mode? And it causes us to think, whether there are some seasonal changes perhaps in how the diatoms are interacting with the environment, or something like the way that Eucampia in the polar regions has a winter and a summer form, or perhaps there are changes in the way the diatom might interact in the environment if it is facing some salinity challenges, for example, if there are changes in this kind of nearshore environment. And another oddity and puzzle is seeing these, like I call it a skin or something that might have been wrapping across these filaments in some way, not really looking like girdle bands, but also showing that same kind of fine perforate structure across here, and then these hexagonal supports or network or something like that. So it's a bit of another oddity. And then also comparing the material from Australia to that of the well-preserved material from Antarctica, you can kind of see how these are still related, spines coming in through here. Some recent work from Japan has been very instructive in helping us fill in some of this missing time interval, right? So that, as well as some work from I've been doing and with Jakob Witkowski and my student Megan Hines in the Canadian Arctic on some samples up there, but filling in this missing piece of time, kind of the diatom dark ages as well. Tsuchiko Shimada here has been working on some material from Hokkaido, and I'm going to walk you through part of this stratigraphic column. It's kind of a composite column, and look at some diatoms from this time interval in the early Campanian, take a step down to this time interval here, and then the oldest record they have down through here. So they're really spanning from Cenomanian up into early Campanian, maybe 15 million years of time, which has been those dark ages that we've had. Also interesting to know within this, and my highlights here have moved a little bit, but this is a deposit that's coming from these cold methane hydrocarbon seeps into the sediments. So they've got these mounds of carbonate material, and that's where they're sampling to recover these diatoms. Again, serendipitous occurrences of well-preserved but cemented material. You can get a look at, put a face on some of these as well. In this time interval, we start to see now triangular shapes, not just the bipolar polar shapes. We start to see the presence of kind of organized labiate processes on these things. We see some more araphids in through here. Of course, some silica flagellates floating around in there as well, and some genera that we know to be into the younger time range as well. Taking a step a little bit farther down, I'm going to show you this figure a couple of times, and we can see carbonate concretions, which is where these are coming from, and they are confirming with more detailed stratigraphic coverage across this interval that there does appear to be this floral turnover coming from the Cenomanian into the Turonian by comparing these two different deposits. I got to go get a power cord or I'm going to be in a problem. I'll be right back.
We're gonna have to order some more of that train too.
Sorry about that. I'm hoping you guys had a chance to talk amongst yourselves. Anyway, but associating this again with a more direct sampling across this time interval to note that there is a transition in the floras. So we're working to try to resolve that a bit better by some material from the Canadian Arctic. This is some material from those deposits, several different sites they're looking at, and this is a bit of a messy tectonic disrupted figure, but these represent different ages of rocks through time. You can see that they're different occurrences of things that are, mapped a bit differently, but going across the stratigraphic column through here on this map, they're sampling different intervals of time. This illustration identifies some newer forms coming in. This one right here is a relic really from the deeper time of the genus Gladius with these spines. And then going into the next work here, coming down to the Cenomanian through here, again, coming from carbonate concretions and ammonites, and then noting in this work that there are connections from this oldest material from this Japanese section to those things, maybe from Antarctica. Other work that has gone on from Russia here, from Tatiana and her work. And I want to identify just an error that Pedro Tapia and myself made before we had good time control. We divide up our rocks and our time based on different diatom events, like the first appearance of this species, up to the last appearance of this, up to the last appearance of this species, up to the first. So we're breaking up time based on events when diatoms appear or go extinct. But this zonation of diatoms and this zonation of silica flagellates that we helped build in parallel, it all needs to be shifted down about that far, right? So we're out by probably 10 million years or so of our age control, right? So it was something that that in the Japanese studies, they also noticed that correlations to their macrofossils that we got it a little bit, maybe a little bit, well, we did get it wrong. Everything was younger than it should be. So getting a handle on time is pretty important. This material from Russia is really exquisitely preserved. A lot of the very early Russian diatom work in the Cretaceous by Juzet and by Strelnikova, came from some of the material and some of this very well preserved material. But of course, back for some of those papers, particularly of Anastasia Juzet's work, they were reported mostly by either not the best light microscope micrographs or from hand drawings. So important to go and try to put more thoughts into that. So one thing we're trying to do within work from the Arctic now, from a place called Devon Island in particular, and some other sites, is also fill in this time interval. But we have the opportunity to do things in a way where we have really good age control. So Devon Island is on the Arctic platform. It sits on top of a lot of Paleozoic carbonate rocks, so it did not undergo much subsidence into deeper depths in the earth, so that it's pretty well preserved. And my colleague Jens Harrell and Wolf Duman had generated this from our data set. We sampled this outcrop every meter, and by doing carbon isotopes and total organic carbon, we're able to match this record with standard global geochemistry to be able to then provide some age control for this sequence. So for example, this excursion up through here, which is ocean anoxic event number 3, is these time intervals. We're just matching up these dates, and then these pluses right here represent where the record from Devon Island is tied to these age bits. So marrying time here and age with our depth in our measured sections, or thickness in our measured sections, and we get this nice linear kind of trend of time. Then my colleague Kevin McCartney, a silica flagellate worker, spent a sabbatical with me working through this stuff, and he looked at the silica flagellates, and he has about 20 different silica flagellate events. These are first appearances when silica flagellates evolve or appear, and some last appearances as well, and they fall within this age line. We labeled these to then try to transfer these to other sites in the Canadian Arctic. A place called Sawtooth Range. It's a little bit messy, but we have some control from the carbon isotope geochemistry, and you can see how the silica flagellate events are somewhat making sense. In this case too, a couple of samples down here, they're in that relative time. So we have the ability from several sections in the Canadian Arctic to now add the diatom work onto this, right? And as a composite of this, with Devon Island representing the kind of the longest record, spanning 94 million to about 84 million years. Hoodoo Dome has 2 intervals of time. Sleeger Fjord has a little bit through here, but from these 4 sections, we get a pretty good composite record with reasonably good age control, particularly for the Devon Island section, to now put these diatom events that we will be seeing into into that kind of stratigraphic and chronostratigraphic time frame, so that we will be able to see the appearance and disappearance of these taxa within this time interval, and help build a timescale using diatoms through these different through this particular interval. If we take a look at material then from the Arctic Ocean, those places I was just talking about, Hoodoo Dome, Devon Island, and others, have some taxa in kind of an interneritic inner shelf environment. Some other studies out here from an outer shelf environment, and then some work out in the ocean realm of the Arctic Ocean realm from Andrew Davies and Alan Kemp provide other information about what was going on with the different environments, from the deep marine environment or pelagic environment, let's say, out to those that are more influenced by the neritic environment. And some interesting work from Andrew Davies and Alan Kemp from a core up in Alpha Ridge, and their approach they have of impregnating these sediment cores with some epoxy and resin, and then looking at them. They identified distinct layers of vegetative cells and resting spores or rhizosolinia type of forms in there, and to be able to see like some seasonality through some of these. And in looking at these in just a broken part of the sediment, you can see the very distinct rhizosolinia dominated material in through here, or hemiolus dominated, or some spore dominated material. So in this pattern of looking at the, I guess, seasonal or sub-seasonal record of the diatom occurrence, they're able to make together some interesting perspectives about what was going on in the Cretaceous in the Arctic through here seasonally, from spring bloom, right, and diatoms through here with the seasonal cycle then into production of the resting spores after the spring bloom dies down, and then with stratification during the summer, that you'd have nitrogen fixing diatoms with symbionts in here operating in this realm, in the stratified water column. And then as you get into fall, then that stratification breaks down and you get the deposition of hemiolus chain forming things or rhizosolinia type forms in there. So interesting to see that seasonal type of pattern and cycle from the Arctic, but from things that are 65, 70 million years old, let's say. So again, that seasonal partitioning is kind of interesting, in a way that we would like to try to work forward to be able to assign or check, validate, hypothesize the different environmental conditions for these diatoms. So from a variety of different sites here, I hope this has given you a bit of a snapshot look at the what some diatoms look like. My postdoc, Jason Conan, is going to be working on some of this material from the older ocean drilling program stuff from Antarctica, and trying to validate this connection between these two different groups. Creagra with these kind of forks, but have the vegetative, or sorry, the rest of the valve is probably a series of girdle bands going down, much like rhizosolinia or eusolinia. And then there are some specimens from the Paleocene, from the fir formation, that actually show this type of a resting stage on the bottom of some of these guys. So I want to try to make that connection, but want to see it within the very unique nature of the record here. When we drilled this stuff in 1989 or 1980, 88, these were all black shales. They were dark. You couldn't see any laminations. They've dried up, and now it's clear that they are laminated. So that material that has the exquisite record of well-preserved, really ancient diatoms, we're going to be able to apply similar kind of approaches that Alan Kemp and Andrew Davies and Jennifer Pike have done by looking at what is in these different laminations to maybe try to understand these really, really old great-grandmothers of diatoms by their seasonal cyclicity and who's related to who, what part of the season are they operating in, and try to see how the pelagic part of the diatom world worked back in the day. There also are some other surprises with more work that are coming in. This diatom right here was quite a puzzle for quite some time. It was originally named Micrampula by Hannah from the Merino Shale, the Maastrichtian deposit that's there. And it was just these balls with a neck, not really clear where they fit in. Some people thought they might be type of a resting stage. Some people thought they might be related to some chrysophycean cysts or something like that. But they were really an enigma for many diatom workers. And Kenza has been working really well. He's quite a skilled SEM tech and just chasing these things down. And that is now resolved by finding that attached to the valve of this diatom are a series and rings of spines. So this genus Micrampula is related to the marine diatom Corythron, with these series of spines that lock into these slots, right? And then they expand outward. So we're seeing two different species of this Micrampula from Hannah, but now shifting its text or assigning its taxonomic position into the Corythrales, and then a second species as well from that same material from Deep Sea Drilling Project Site 275. And it's kind of cool as well to think that these spines, which are attached on these hinges or sockets through here, right, that they also go up along this space, and then up here are these costate that also keep them organized. So just kind of wrapping up here, it's really important to tie the morphological record of the change through here of diatoms with the molecular record, and then also to marry it with the stratigraphic record that we have from these different glimpses from some of these really good occurrences, to be able to assign ages for the points potentially of the divergence of these, to be able to calibrate, this molecular time clock or molecular tree through there. Anyway, that's my shtick for today, my spiel about these great grandmothers. I hope I entertained you. I hope that you have you might have some questions for me in all this, and thanks for your attention, folks.
All right, thank you so much, David. That was wonderful.
Thanks.
I just posted in the chat. Feel free, if you want to, you can just type your questions in there, or we've got a small enough group today. I think if you just raise your hand, we can call on you and also ask questions that way. Just as we get started, Dave, and wait for a few things to pop in here, I'm kind of curious. Can you give us a sense? You talked early on about pyridization and finding these things in macrofossils. Can you give us just a sense of sort of your best case and worst case scenario of preparation that has to go into these things? Is some of these things mix them with a little distilled water and put them on a cover slip, and others are insane?
Yeah, some is I'm just too old an individual to mess around with too many muddy things anymore, or things that are just ratty. You have to sometimes, but there's enough out there that still requires good work on some of this. But sometimes it can be as simple as just a smear slide or putting it on some water. Other times, peroxide certainly helps, and hydrochloric acid if there's any kind of calcium carbonate cement. But you kind of want to see the calcium carbonate cement in there again, because you've locked things in a better way. One thing Jason Conan is doing is he's also following up on this work I did across the Cretaceous Paleogene boundary, across the KT boundary, basically to see that extinction. That was all from calcareous concretions, carbonate concretions as well. And he's trying to look at it and sample from the outside of the concretion to the inside to see if there's some progressive increase in preservation. But again, a lot of times you get lucky. For that project, we're probably about 30%. 30% of any sample you look at has diatoms in it. The other 70 not. Sometimes it's much less. From the material that Megan Hines is focusing on and Jakub Witkowski focused on, and I'm working on too, is from the Arctic, Canadian Arctic. Maybe in general, those are like 8% of all the samples you look at have some diatoms in them. But from this Devon Island section, which is just beautiful, it's 100 meters of sampling every meter. It's all rich and really good. So that's going to be kind of like a Rosetta Stone, if you will, of figuring in some of that. The Japanese material, they're reporting basically on just one sample in each of the materials they've looked at. So I'm sure they're probably in that same situation. If you look at a lot and then you get lucky on some of them. So, but the preparation technique is pretty simple. Diatoms from the Canadian Arctic were originally found by a person, Karen Chin, who's working on coprolites, just from looking at thin sections of the coprolites and said, hey, what's in that stuff? We then tried to prepare the coprolites directly, and they were just carbon shadows of what was there, right? So whether the coprolite formation got rid of the silica or whether they actually captured something, I don't know. I think I raised this at the NADS meeting as well, that issue. But yeah, sometimes you get lucky, and sometimes I'm surprised that they all went away. But anyway.
All right.
Good. Sorry,
Great,
long
thanks.
answer there. Thanks, Mark.
Thanks. Sarah, why don't you've got your hand up? Why don't you go ahead and ask a question, please?
Yeah, David, thank you for just such a fascinating talk. It's
Oh, thank
always
you.
so wild every time I see what you're
Okay.
working on. And one of the things I'm always kind of curious about is just silica concentrations and how much that impacts, or if you look at that at all in terms of how much silica is available dissolved at any particular geographic time place, and especially when you're talking about some of the non-marine forms of what kind of silica availability would be there and how that ties into the external ornamentation.
Yeah, and preserving all of that stuff as well, right?
And preserving it, yeah.
Even, I mean, it's often like a lottery, of what's preserved. If you have a lot of diatoms around being produced and a lot being buried, some are going to be dissolved, and that's going to help some others be preserved. There appears to be, although I don't recall anybody really quantifying this, but an association with volcanic ash and volcanic material. So whenever you have volcanics in your sediments, you're also providing some bit of a buffer. But I think it's going to be like anywhere, in my experience, in the marine realm mostly, is wherever you have a lot of silica, wherever you have upwelling, wherever you have that growth, you're going to have diatoms preserved. Even though diatoms were in the water column, they aren't going to make it to the seafloor, if the oceans are undersaturated as they are, right? So you've got to dump a lot.
But I guess what I was really trying to get at is how those silica concentrations influence the form of the spines or the extra valve, the silicification of the valves.
Yeah, I get I understand what you're saying. I don't know. There's not a whole lot of variability as a result of the dissolution. I mean, I think even with the modern diatoms, if there's enough silica, they're going to make their forms. With some other siliceous microalgae, like with ibridians, for example, or with some silica flagellates, they will change their form. Radiolarians do this too, based on how much silica is there. But I don't know if your guys' experience in the non-marine realm, whether there is that much of silica availability changes their morphological expression. I tend to think that they're going to pull out the silica if it's available. They're pretty good at it, right? And then they're going to do what they always do, right? I don't know, but do you guys have that much variability in the non-marine realm when you have more silica or less silica? Is there much of a morphological change?
Yeah, well, Mark can talk about that.
Okay, sorry.
Yeah,
for sure.
yeah. I mean, we do have some, we do have some evidence of that. Even recognizing different forms of diatoms, so treating them as forms based on essentially their silica supply availability.
Okay, cool.
But Joe, go ahead and ask your question, please. Unmute.
Hey, Joe, how's it going?
Hey, David, great talk.
Thanks.
I'm glad you touched on possible preparation methods, because I've got a box full of the
Oh,
Hell
great.
Creek formation.
Okay, awesome.
So our dinosaur paleontologist here at the museum has been working on some marine transgressions right around the Cretaceous impact event.
Cool.
So it's a little lower. So I guess what I'm asking is, it looks like I need to expand my thought of what I might see under the microscope and be able to identify as a diatom. You know, they look pretty different. I've worked on some 3 million year old diatoms and some 12 million year old diatoms, and these are, you know, 65 plus. Just wondering if you have any advice dabbling into this realm.
Yeah, so at first, the sample you had there seemed to be a pretty big sample, right? Which is good, but I would tend to go more for lots of small ones, right? It's kind of a shotgun effect, right? An approach that is going to be more successful than a lot of big material, a lot of volume from any one interval, because you're going to get lucky, and it really that's that serendipity bit. And also, if you look around once you do your initial preparation, if diatoms are there, you're going to see fragments of them. You're going to see traces of them, right? So I wouldn't spend a whole lot of time looking through things that are just barren. I used to do that, but again, I'm an older man now, so I have my time is more precious. And it just turns out that if they're there, they're there, and then you can try to then concentrate them, right? But I also, when you said, I think, Mark, is it just a little bit of mud on a some distilled water and see where you go? I would start that way, and then I would
Really? Okay, cool.
well, just to see if there's anything there. Otherwise, you're going to waste a lot of time for stuff that's just not productive, But then go to those where there is a trace, and then work on that in a much more comprehensive way. And I'd be happy
Cool.
to share some methods with you, be it from floating diatoms by density or by different settling techniques. But if you do have diatoms in that sample you just showed me, it's great to have that great volume, because not everyone's going to be well preserved, right? But you can concentrate them by size, you can concentrate them by settling to bring them up. So
cool. Yeah, that's great. I've got plenty of material and collected from different places. So yeah, getting a sense to just smear slide it, see if there's anything there, and then I can do some heavy liquid if necessary or whatever. But if I find anything, I'm going to reach out, because I'm not going to know what any of it is.
Okay, no, do that for sure. And if anybody was looking for a big project, or an early career scientist looking for something that wanted to try to address this, there's a wide range of potential new projects to come, just by sampling from museums, just by sampling from geological survey collections. And again, it's a shotgun approach, but you never know if a formation is going to be productive. But if you can go and they'll let you take a little bit of a cemented, bit of a concretion off of an ammonite, or drill into the central void, and then you just going searching, and shopping, if you will, and then you'll know, hey, that formation could be productive. So I think a lot more things could be known just by sampling those things people have already collected.
Yeah, that's great. That's the approach here. Thanks.
Okay, good. Yeah.
All right. Shopping for diatoms. I'm going to remember that. So, we got a question in the chat, David, from Andres. Pixidicula is one of the oldest diatoms ever reported, bringing up that Roth plus 1896 publication from Jurassic sponge reefs. And you had indicated that sample was lost during World War II, and she is wondering whether there's been any attempts in resampling this site, and have you ever considered this possibility? And maybe we just need to get Carolina in for a talk.
Yeah, she might be able to say something about her search for that stuff. But I think Benda back in the 70s and 80s looked hard and long at wherever those things were supposed to be. Even some silicious sponges from Poland. I think Vladimir Nikolaev got a bunch of samples from some folks there looking for them as well. But yeah, no, they're fleeting. They're whatever they are, and they're just these hand drawings, but they kind of fit, to be this capsule shaped thing, a central dome. I once for a while thought they might be, not even silicious, but from the description that Rothplatz did. I think Linda Medlin had that translated recently, and it appears that they are residual from a hydrochloric acid treatment, and they appear to be in there, but that's just what we've got, right? Yeah.
Okay, thanks. Layla's asking us, have you seen major extinctions of some of the genera at the end of the Cretaceous, and how do you explain that diatoms did not exist before the Jurassic?
Yeah, no, really good couple of questions there. So the only site where we actually have a record across the Cretaceous Tertiary or Cretaceous Paleogene boundary is this work that I did long ago on Seymour Island. And this is something that Jason Conan is also focusing on for his postdoc, is to resample that, to look through the macrofossils through that. And actually we're funded, Tom Tobin's the lead PI, we're funded to go to Seymour Island with a drilling rig and sample right across the boundary at 2 different sites. It'll be pretty hard work, but really heroic stuff of landing craft, storming the beach, off of an icebreaker, right? Pulling a drill rig with a small quad vehicle. And anyway, but so there's a chance to go back through that. But no, the answer to your question is the diatoms seem to go across that unscathed. Some of the spiny things like gladius, there are probably 2 genera that go extinct, but for the most part, they just continue right through. And the thought is that maybe the diatom's ability to, form resting spores or some kind of stages that could live through multiple cycles, that they seem to survive. But other fossil groups, of course, in the planktonic forams, calcareous nanofossils, silicoflagellates, even from that same study, they get nailed across that boundary. But for some reason, diatoms do not. So that's an interesting kind of a study as well. The work that Tom Tobin and Jason and I are doing, Tom Tobin, who's a macropaleontologist, he's actually seen a precursor extinction, they call it, a little bit before the Cretaceous Paleogene boundary. So it's trying to see whether the diatoms or the microflora show any hints of that, earlier extinction that is hinted at in the macrofossils. And then, yeah, sorry, could you remind me what the other question was in there? The second one.
Why did they not exist before the Jurassic? Why did diatoms?
Yeah. So that's hard to know. Maybe they were still around, but they were in different environments, right? For one, maybe they were terrestrial. I've always been amazed, and I almost pulled this into this talk, but I thought it would be too much. But aerophilic diatoms like Orthocera, for example, those things look like these really ancient diatoms. So I had hypothesized in a paper with Vladimir Nikolaev that maybe that archaic interval of diatom evolution is still with us. It's just, that group just stayed in the terrestrial realm, and the extinction at the Cenomanian-Turonian boundary was a marine extinction. But many of those members of that earlier group, they survive today in this aerophilic environment, which also helps to maybe feed a thought that diatoms started there as well, right? So that's maybe a bit outrageous, but I don't know. that's where I'm still plugging in to think they have a long record from that. It could be that diatoms did not start adding silica until later, but they were still around, because the molecular work shows they should be older than our fossil record is showing us. But all that depends on how well we can calibrate the molecular trees, to when those branches are occurring. So that's why the stratigraphic record that's coming from Devon Island and from Japan, that's better dated, will provide in the next decade, hopefully, much better anchors to the molecular clock trees. Right.
All right. Well, we reached the top of the hour. I want to first say thank you, David.
Welcome. Thanks.
Wonderful presentation, and just
Thanks.
such a neat. David Burge had noted earlier that we can appreciate modern diversity, but this is the temporal diversity that we're really getting when we see this kind of thing. I want to remind everyone that in 2 weeks we're having another Diatom Web Academy that's going to be covering. Sylvia Lee is going to be covering our taxonomic certification phase level 2 exam, talking through that. And sure appreciate everyone joining us today, and just again want to thank David for presenting his work to us. Thanks, everyone.
Thanks all. See ya.
Thanks, everyone.